Good morning, students. I hope you all are safe and healthy. Today, I'm going to talk about topic gene therapy. Already, lot many topics of genetics we have covered, and now I believe that you all know the basics of the genetics. So, by that, once you know the basics of the genetics, understanding gene therapy will be comparatively easier for you all. Now, gene therapy, which is one of the very vast topic, vast in sense because it has lot many methods to apply for the gene therapy, to do the gene therapy, and also there are various sections out of it. Means first you have to identify the disease, which disease is related to the genetic disease, find out the what particular gene in that, and then you identify which technique to modify that particular gene or to replace that particular gene. Like that is the process. So lot many methods will come, and after that, after discussing the methods, we will talk about the various applications of the gene therapy. So I hope you may get this topic very well. So let's move on to the introduction part of it. First, what I am planning, I will just brush you up with some little things what we have discussed already. Right. So here I will just talk about the gene a little bit because we are going to do the gene therapy. Gene is termed as a biological unit of heredity. Already discussed about the gene a lot. The genetic message is encoded by subunits of the DNA called nucleotides. There are approximately 3 billion pairs of nucleotides in the chromosome of a human cell. Now you can understand, if there are 3 billion pair of nucleotides, among these nucleotides you are going to find a one gene, right? And in this process, you remember we have discussed about the gene mapping. So gene mapping helps in to identify or to map out all the genes. Then you can identify that particular gene which is responsible for causing that particular disease. Despite technology breakthrough in viral biotechnology, out of thousands of gene therapy clinical trials that have been initiated, only very few are now reaching to regulatory approval. So this is the truth of the gene therapy, right? Because there are a lot many ethical issues, there are a lot many regulatory issues. So unless until these issues are not get satisfied, we cannot initiate any of the therapy process, right? So that's why very few clinical trials go into the another phases. Okay. Here an experimental procedure aimed at replacing, what the aim of the experimental procedure for gene therapy. So this is talking about that. So an experimental procedure aimed at replacing, manipulating, or supplementing. So three things are here: replacing, manipulating, supplementing. So replacing means you have to replace that particular gene. How? With what? With a manipulated gene. Okay. And supplementing, and that is manipulated with the supplemented, means that manipulation has been done by what the we have supplemented some information into it, right? Non-functional or malfunctioning genes with healthy genes. So like that we do these things, replacing, manipulating, and supplementing, and then we, that exchange we are doing with that particular gene which is a faulty one or which is a uh, diseased one. Transfer of genetic material to cure a disease or at least to improve the clinical status of a patient. So, what is the aim of the gene therapy? Aim is to, improve the clinical status of the patient, or possibly first is the to cure the disease. If cure is not possible by some reason, we have, at least we can give some better clinical status for the patients. Here, insertion of a functioning gene into cells to correct a cellular dysfunction or to provide a new cellular function. So this is also the part of the aim itself, that we are inserting a functioning gene into a cell For what? To correct a cellular dysfunction. Because in the disease, because of the gene was not behaving well or not functioning well, the functions of the cell is get disrupted. And that's what it causing the disease. So that is our purpose. To that particular gene which is not functioning well, we have to replace it and provide the new cellular function to the cell. For example, diseases such as cystic fibrosis, combined immunodeficiency syndromes, muscular dystrophy, hemophilia is a big list where the gene therapy has been tried, and in some cases it is successful too. So, what are the content for this presentation I have brought you? I brought some brief history. I have a brief history of gene therapy. Means you have to know when this all have been started, who has started, how the progress has happened. So just a little idea about it. Introduction, brief history. Introduction, approaches and types of gene therapy. Uh, applications of gene therapy, gene therapy in disease, ethical issues, development and future prospects. So these are the content what I am going to deliver by this presentation. These are the references what I have used. After this slide, I will take you to the journey of these three research papers. These three research papers are enough to explain you everything about the gene therapy. So first one is the introduction to gene therapy. Second is the gene therapy a comprehensive review, and another is also a kind of review is giving the information. So from here onwards, I will take you for that, and I'll open the PDFs, and then I will explain you all of these processes which are happening with this. So I'll take you to this first part of it, where we'll discuss about the history of the gene therapy. Exactly, around 30 years ago, there was one scientist known as Martin Klein, and around 1979. So he has, initiated one gene therapy, but that was very premature human gene therapy attempted. So actually, that time it started. What they were trying to do, they were trying to treat thalassemia patients through an ex vivo beta globin gene transfer protocol in the bone marrow. So that was they were trying on that time, but that was a very premature initiation because it was the first time. As the protocol had not received any other mandatory approvals, the study promptly terminated. So, like that, the study has not gone for the next level. Now, there are some committees who have acted on this. DNA Advisory Committee, which is called RAC at the National Institute of Health (NIH), was urged in 1980 to expand its regulatory function beyond recombinant DNA experiments so as to include human gene therapy studies. So initially initiated it on that time, but a lot of, as I told you in beginning, that lot many ethical issues were there, lot many, and nobody was knowing exactly that what it will come out as if they try in this way. So in 1989, Rosenberg initiated the first RAC approved gene therapy. So that time RAC has given the approval and they started the clinical trial. That was like a gene labeling process. They were labeling a particular gene and then they are trying to remove it off. Targeting a neomycin resistance gene transfer into tumor infiltrating lymphocytes using a retroviral construct. So what they have used? They have used a retroviral DNA to transfer their gene into the what called the human genome, where they want to replace it. For the treatment of what disease they have targeted? Metastatic melanoma with interleukin 2. Effectively, a therapeutic gene in clinical trial took place in 1990. So you can say these kind of efforts has been initiated from 1979 to 89, and later in 1990, one effective therapeutic gene clinical trial happened. And what they were trying to treat on that time? SCID, that combined immunodeficiency disease, by transferring the adenosine deaminase gene into T cells using a retroviral vector. So this way they have initiated on that time. We'll see some more basic concept about this. The concept of gene therapy is relying on gene intervention. So what ultimately you are doing? Gene intervention means what and where the gene is intervening, and you are intervening that particular gene's function. So like that you can say. The virus-derived gene delivery vectors were thought from the beginning to be optimized biometric vehicle. The problem is, like, when you want to modify the gene, or you can say that you want to replace a gene from another gene, so you need some vector, some vehicle, some transport system by which you can take the gene from outside and then you insert into the body, and then that, so by which you will insert, by who will take this gene into the human genome, right? So that's what the vectors were used, right? Viral vectors have been used. Since they are also evolved under a very high selection environment of infectious agent, humans are also naturally equipped with very sophisticated defense system. And you all know that whenever any foreign substance enters in your body, your defense system get activated and try to destroy that. So this was another difficulty was faced by the scientists when they were trying to insert something from outside, right? So these defense systems, which are often specific to higher primates, cannot be ignored in the context of a gene therapy. So you cannot ignore them. Especially when it comes to use of a natural human-derived viruses. Other hurdles are the active virus loads and the amount of virus particles to be used to achieve therapeutic effects. So these were the difficulties faced by the scientists during that time. As foreign DNA cannot stay freely in dividing cell, it does not get associated with the host DNA replication machinery. So it cannot, if you insert a foreign DNA, it doesn't mean it will go and incorporate into your DNA replication process. So there we need some other methods to make this DNA to be the part of the host DNA. On one hand, one has engineered integrative vectors enabling the therapeutic gene to be integrated into the host. So likewise, same time the vectors discovery was going on and find out the particular vector, that vector can carry the therapeutic gene to the host, thereby enabling long-term expression potential. So what another problem is that if you need to incorporate that particular gene is nothing but a part of a specific part of a DNA. So all DNA, the part of the DNAs go for the replication process, go for the what called the they involved in their gene expression as well by the protein synthesis. So what happens if this particular DNA what you have taken from some other virus, if it is not having that high expression, then you won't get the result out of it. Means that that particular replacement what you have made must be equally or potentially expressive. It should give the desired gene products. If that is not happening, the purpose is not getting solved properly. So here what happened, they enabling the long term expression potential. So that's why for getting more expression of or more protein synthesis, you can say, they are using oncoretroviral or lentiviral vectors. So it means they are using that kind of uh, uh, genes, or you can say the viruses, which are able to produce high expression. So they have used the oncoretrovirals. A major drawback in this random insertion into the host genome that can lead to serious adverse effects. So that's always there when you are jumping into the unknown area. The chances of adverse reactions are severely, because we don't know which way the body is going to react. In addition, the route of administration of a therapeutic principle can have major consequences. So, means the route of administration, which way, suppose that you have made all this process done and you got medicine which can do this process, then what should be the route of administration, right? So, that's the one difficulty is always there, because you don't know what will be the efficacy, what will be the safety, by which route, what kind of efficacy safety you are going to get. So these difficulties will be there. Routinely one classifies gene therapy protocol into three main categories: ex vivo, local in vivo, and systematic in vivo administration. So like that, these methods have been utilized in this process. So now, as you have seen, the vectors, which are very important carrier molecule for you, which are very important vehicle for doing this process. So we have to see what are these vectors and how they are doing it. So I'll take a little detail about the vectors here. Facilitating the transfer of genetic information into a cell are vehicles. So these are which are facilitating the information into a cell are the vehicle, simply called as vectors. Vectors can be divided into viral and non-viral delivery systems. The most commonly used viral vectors are derived from retrovirus, adenovirus, and adeno-associated viruses. Other viral vectors that have been less extensively used are derived from herpes simplex viruses, vaccinia virus, or baculovirus. So they are also there, but not very much used. Other than the viral vectors, the viruses are not only the medium for transferring the material. We have non-viral vectors as well, and they can be either plasmid of DNA, which is a circular of double-stranded DNA that replicates in bacteria. So we are taking the plasmid from the bacteria. So not always the viruses, but the bacteria are also contributing for this gene therapy by giving their plasmids or chemically synthesized compounds that are resemble oligodeoxynucleotides. It means not only the bacteria and viruses, you have some synthetic compounds also, right? But they we are preparing them which has some resemblance with the oligodeoxynucleotides. Major considerations in determining the optimal vector and delivery system are the target cells and its characteristic. That is the ability to be. So you just remember here there were two considerations. We must consider why and how we select the particular vector. So number one, the target cells and its characteristics. So the selection of virus or selection of vector or selection of carrier, what you are choosing, you must keep the which cell type and what is the characteristic of that particular cell. And also the you see that virally transduced ex vivo and reinfused to the patient or not, and how it's doing. Another one is the longevity of expression. Once you are using that particular virus or particular vector, right, that has to have that capability to keep the expression for longer, right? The size of the genetic material to be transferred. Another very important point is the size, right? So what size of the viral vector you have means what size of genetic material you have. On that basis you can select, means that much material you have to find out the correct size of the transport medium. Okay, so like that you have this thing here. One table I have given. This table is talking about the various viruses and their characteristics. Here you can see the size and type of genome of the viruses, which viral proteins they are producing, physical properties what they have, and in which diseases they are caused, which diseases they are involved in. So here are retroviruses, adenovirus associated viruses, herpes simplex viruses, vaccinia viruses, baculoviruses, and all. So you can go through with the table and you can understand how these are behaving. Now we'll see the viral vectors in little more specificity and little more detail. So viral vectors, after this viral vector, we'll talk about the non-viral vectors as well. Now we'll see first what the viral vectors are and what the characteristics they have. First is the retroviruses, a class of viruses that can create double-stranded DNA copies of their RNA genome. You know what the specificity retroviruses have already, we have discussed in the previous chapter. So you know they are the viruses which are involved in the HIV infections, right? Where they are doing a wonder to convert they make the DNA by the RNA, right? So that's why they are very special kind of viruses. And you know they are having a lot of very good expression ability as well. So we'll see how they are doing it. One of the problem of gene therapy using retroviruses is that the integrase enzyme can insert the genetic material of the virus into an arbitrary position. What does it mean? Means once you are inserting the virus with the gene loaded into it, gene of your interest loaded into it, and sending into the host, then what is the problem happens here? The integrase enzyme which is doing that job for these viruses, they will place that genetic material into the human genome in an arbitrary position. Means it's not taking that genetic material into the specific position what is your desired position. So that's one of the difficulty creating. It randomly inserts the genetic material into a chromosome. If genetic material happens to be inserted in the middle of the one of the original gene of the host cell, the gene will be disrupted. Means suppose that you have inserted this particular gene what is loaded into this particular virus and to a normal gene which is not a gene of your target. So what will happen? That gene which is a normal gene and functioning normally, and you have disrupted it by this, now the another that gene is also getting malfunctioned. Now that will create more problems. So these kind of problems being called as the insertional mutagenesis. Maybe these can cause the in some kind of mutagenic conditions or some kind of tumor they can create through the insertional disturbances. So for example, if the gene happens to be one regulating the gene which you have disturbed is the gene involved in the cell division process, then what will happen if you have disturbed it? Now that uncontrolled cell division will be the result, right? That tumor or the cancer will be the result. The problem has recently begun to, and now they have got some solution for it. For using the zinc finger nucleases. You remember the zinc fingers you have seen in the cell communication pathways. The zinc fingers are like an anchoring site, or they help in to anchor that thing to in the genome. So that's why the zinc finger nucleases have been utilized, as well as the another method they are using the beta globin locus to find out the correct beta globin loci control region to direct the site of integration to specific chromosomal sites. So this is the way. These are the difficulties, and this way they can solve it. Now the adenovirus, the another one, which is also the good virus for the vector. A class of virus which double with double-stranded DNA. So they are having their own double-stranded DNA. They are not converting RNA into double-stranded DNA. That can cause respiratory, intestinal, and eye infection. Means they are the viruses which are usually giving the common cold kind of conditions. When these viruses infect a host cell, they introduce their DNA molecule into the host, like normally they do. You have already seen in antiviral chapter how these viruses infect the what is the infection you know pathway for them. The genetic material of the adenovirus is not incorporated into the host cell's genetic material. The DNA molecule is left free in the nucleus of the host cell, and the instructions in the extra DNA molecule are transcribed just like any other gene. So what they do, the DNA molecule is left free in the nucleus of the host cell, and the other DNA molecule, which is a host DNA molecule, will utilize this or treat it like any other gene and will go for the transcription process. The vector system has been promoted for treating cancer of liver and ovaries, and intended the first gene therapy produced to be licensed to treat head and neck cancer. So what that known as gendicine. So this is an adenoviral product, right? So here I have given a one small summary of this whole process. So just go through with this. So you can find out that why and how these things are being done. So in that whole process, the most gene therapy studies a normal gene is inserted into the genome to replace an abnormal disease-causing gene. So that's what they do. A carrier molecule called a vector is used to deliver the therapeutic gene to be patient's target cell, that you have understood. Currently, the most common vector is a virus that has been genetically altered to carry normal human DNA. You have seen that. Which are these viruses? Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. Scientists have tried to, you know, take advantages of this capability, and like that they have used viruses, and they have using it for delivery process. Now the another virus which we are discussing here is adeno associated virus, AAVs. These are one of the most promising potential vector, is a recently discovered one, and which infect a broad range of cells, including both dividing and non-dividing cells. AAVs are small viruses from the parvovirus family with the genome of a single stranded DNA. It can insert genetic material at a specific site on chromosome 19 with a near 100% certainty. You know, this kind of virus, if you get who has that specific action, then what else we need for this purpose? Researchers believe that most people carry AAV which do not cause disease and do not provoke an immune response. So like that, if your body already know this virus, then the defense will also be silent, and your purpose will be solved comfortably. It is now being used in preliminary studies to treat hereditary blood diseases, hemophilia, muscle and eye diseases. The chief drawback of AAV is that it's a small, carrying only two genes in its natural state. So this is one of the drawback what they have. They are very, very small. So you have a limited in the loading size. You can't load a much amount of the genetic material. Next is the herpes simplex virus, HSV. This HSV is a class of double-stranded DNA viruses. As a class of double-stranded DNA virus that infect a particular cell type, neurons. So they are infecting neurons as well. Herpes simplex virus type 1 is a common human pathogen that causes cold sores. In a human neurotropic virus, it's a human neurotropic virus, right, which is going to cause the neurological genetical diseases, which is mostly used for gene transfer in our system. It has a large genome compared to other viruses, which enabled scientists to insert more than one therapeutic gene into a single virus, paving the way for treatment of disorders caused by more than one gene defect. HSV makes an ideal vector as it can infect a wide range of tissues, including muscle, liver, pancreas, nerve, and lung cells. The wild type of HSV1. Here, one more specification, one more type of HSV1, which is a wild type of HSV1. Virus is able to infect neurons which are not rejected by immune system. So this is the benefit what we get out of it. Now non-viral methods. Let's discuss about the non-viral methods. So why we always depend on the viruses? We have some non-viral methods as well. So in comparison with virus-derived vectors, non-viral vectors have several advantages, of course. They are such as the safety of administration without immunogenicity, almost unlimited transgene size, and the possible of repeated administration. You can use it many a time. Non-viral gene delivery system generally consist of three categories: naked DNA delivery, lipid-based, and the polymer-based delivery. Naked plasmid DNA. First, we'll see the first one, which is called naked, which is the naked plasmid DNA. The simplest technique of non-viral gene transfer is the use of so-called naked DNA. So, how the approach of this one? Like naked plasmid DNA transfer method, wherein a cytotoxic T lymphocyte antigen 4 immunoglobulin gene was delivered using a naked plasmid DNA. This is an example given. Naked DNA was used for anti-angiogenic therapy, where the fetal liver kinase 1 gene was delivered. Another very important and very promising one is called the cationic lipids, right? So you might have never heard about it, the cationic lipids. So how they are used, we will see. The cationic lipids. Now the cationic lipids Cationic liposomes are an important class of compounds suitably for carrying negatively charged DNA. You know what is the benefit of the using lipids for the carrier for as a carrier? Because nowadays lot many drugs are also been entrapped under the various type of lipids and been used as a carrier molecule, because lipids are having a very good affinity with our body system, because every cell and its plasma membrane is having the lipid affinity. So that is what the lipid can be one of the potential target for this. So there are at present several commercial transfection reagents that are based on cationic lipids, like the lipofectin, DOSPA, DDAB, neofectin, transfectam, etc. So these are all used for this particular purpose already. Cationic lipid consists of a positively charged head group. This is nothing a very new information. You know that how the lipids in the plasma membrane looks like. So they are also having the similar kind of structure. Cationic lipid consists of a positively charged head group, a polyhydrophobic tail, and a linker connecting the head to the tail group. The charged head groups are usually quaternary amines. Tails are saturated or unsaturated alkyl chains or cholesterol groups. Cationic liposomes, in contrast to neutral and anionic liposomes, which need DNA implementation into the vehicle. Cationic liposomes naturally create complexes with negatively charged DNA. So they are very well receiving the DNA when we load into it, and same time they are well received by the body when they are enter into the body, and will get more convenient penetration, more convenient reception by the body, and so they can enter into the nucleus comfortably. Their positive charges, moreover, allows interactions with the negatively charged cell membrane, and thus penetration into the cell is permitted. One more is there that you can call it as a polymeric gene carrier. So the synthetic polycationic polymers, these are the synthetic compounds, right? Synthetic polycationic polymers have gained wide attention as non-viral vector for gene delivery. Examples say are here like PEI, polyethylenimine, is more appropriate as it's as a set a gold standard for non-viral gene delivery. Their ability to condense large DNA molecule and eventually leading to homogeneous spherical molecules of 100 nanometer size. So they are, their ability is to condense a large DNA. So this is one of the very important characteristics they have. They make it up to the molecular size of 100 nanometer size, right? As they are capable of transfecting into cells, affecting for both in vivo and in vivo, efficiently for both in vitro and in vivo. The other synthetic polymers showing promising results in gene delivery are poly-L-lysine. So you can see here, uh, this you have seen like PEI has been used here, poly-L-lysine, as well as you can find later the imidazoles, which are also been the synthetic compounds for delivery process. So the other synthetic polymer showing the promising result is the poly-L-lysine. It is one of the first polymer to be studied for non-viral gene delivery because of its peptide nature. That is, it is biodegradable and hence it is more suitable for in vivo use. Imidazole containing polymers have been reported to have efficient transfection properties as well. Another example is also there called amino methacrylate polymer. So this is all about the viruses and non-virus vectors. And once we understood the vectors, so we have understood one component of the in the process of gene therapy. Now, in the next section of this video, we will discuss about the different technologies which are used for the gene therapy. Of course, these technologies are going to utilize these vectors into that, and by various methods, they will load the desired gene into these vectors, and then they will allow these vectors to go into the body, and then the further removal or the modification of the host gene will be happening. So in the next part, we will discuss about the technologies, as well as we will see once we do these processes, what are the wide applications of it in the medical science, in the therapeutics, or in other areas of the daily life. So now I would like to say thank you all of you for listening the video gene therapy part 1. In the second part, we will see the technologies and the applications. So, thank you all of you for learning honestly.